Good afternoon. I'm very happy to be here today to show you our work on our human stem cell-based retina chip and how can it be used as a screening platform for drug development. The retinal tissue is very, very complex, and it's very challenging to design a chip that meets the requirements of the layering of the retinal tissue. That's why we thought that maybe we can merge the organoid technologies with organ-on-a-chip technologies. Why? Because the retinal organoids are incredibly well differentiated when we compare with other tissues like liver or heart. They present already the most relevant retinal cell types, including Muller glia, photoreceptors, cones and rods, ganglion cells, bipolar cells, amacrine cells, and horizontal cells. This work has been, the development of these organoids has been done in the Liebau lab, also in the University of Tübingen, and we work in collaboration with them for a really long time. So, more important than having all of these cell types is that they have a physiological layering. So the outer side of the organoid will be completely filled with photoreceptors, while cells like the ganglion cells would be in the inner part of the organoid. More older and more developed organoids would at some point be relatively hollow. So that's how our retina chip looks like. So we created a microfluidic platform where we could place 4 organoids that would sit within an hydrogel on top of the retinal pigmented epithelium called RPE. And then the RPE are seated on a semi-porous membrane which is perfused beneath with a vasculature-like perfusion. First, we characterize the RPE. So the RPE on the chip have shown to recapitulate the physiological structure of the RPE in vivo, meaning being pigmented, confirmed bilateral microscopy, to form microvilli, and to be polarized. So they were able to secrete more VEGF towards the direction where the endothelial cells would be, so towards the vasculature side. When we placed the organoid on top of the RPE, embedded in the hydrogel, we were able to have a defined spacing of circa 5 micrometers between the organoid and the RPE. And this is very important because, as you see here in green, the organoid photoreceptors outgrow towards the RPE, and this space is needed. Otherwise, the organoid could disintegrate. This is how they look in detail, the outgrow of the photoreceptors on the chip after 7 days. And we could show that a chip cultured without RPE or an organoid on a dish are not able to produce the same amount of outer segment-like structures as when the RPE and the organoid interact with each other. So now I need to recapitulate what's the main function of the RPE. The RPE function is to supply the photoreceptors with nutrients, to get rid of metabolic waste products, and most important, participate in the recycling process of the photoreceptor outer segments. So to verify if this was happening on the chip, we labeled the photoreceptors with PNA lectin and were able to do live imaging over a period of 6 hours, and we could actually see this internalization. The PNA lectin is told to not be very specific for the photoreceptors and outer segments. So to be completely sure, we confirmed the staining with rhodopsin antibodies after fixation. And we were able also after 1 week to identify the outer segment-like structures inside of the RPE by electron microscopy. So we were able to recapitulate the in vivo segment recycling processes. So our chip is already characterized well enough in our perspective. There's always more to do, but we thought we could already go into pharmaceutical applications. So what we've done first was to focus on drug safety and small molecules, where the small molecules are perfused through the vascular-like channel with a flow rate of 40 microliters per hour. We selected the chloroquine, which is a classical retinal toxicant. It's also an anti-malaria drug. And we used 2 doses, 20 and 80 microgram per milliliter, and used traditional propyliumidide to identify nucleus of cells that have higher permeability, so they're either apoptotic or dead. And the higher concentration of chloroquine led to a much higher PI intensity. And looking into the mechanism of toxicity of chloroquine that is known to induce lysosomal accumulation, we were also able to observe that. So we could have a mechanistic insight. Exactly the same experiment was made with lower doses of chloroquine, but over a longer time, and toxicity at lower concentrations was observed for both concentrations after 3 weeks. So the next step would be to see if the system would also react to light, which is the most important function of our retina. So we use the harsh condition of chloroquine, so 3 days, 80 microgram per milliliter, and use light amounts that are considered to be toxic, so exposure for 2 hours to 5,000 lux. And we could observe that the chloroquine did not change the ATP content of the retinal organoid, but the combination of light and chloroquine did. So we could recapitulate the retinopathic side effects of chloroquine. The next step would be then to study other drug delivery systems with our retina chip, here with a focus on gene therapy. The eye is very easily accessible for local drug delivery, and the main drug delivery systems for the retina are subretinal and intravitreal. Intravitreal is a very simple procedure where a drug can be inserted into the humor vitreous, but the inner limiting membrane is a big barrier, so you can put the drug there, but the drug will not reach the retina easily because it has to go through this membrane. Subretinal injection means adding a drug directly on the photoreceptors, which sounds okay, much easier, but the risks of this intervention are much higher. One of the major risks is the detachment of the retina. And why gene therapy and why AAVs? A lot of retinal diseases correspond to a lack of expression of a specific protein, and using AAVs to lead to the local production of this protein is a very tempting pharmacological approach. And AAVs, and especially recombinant AAVs, have an excellent safety record, have a high transduction efficiency, they allow long-lived gene expression, especially in post-mitotic cells and cells in tissues with low regenerative rate. And most importantly, they have episomal expression, which means there's no integration into the host cell DNA. Problem is that AAV cell tropism is very cell-specific and species-specific, and kinetic information of the infection is very important for decision-making. Still, in Germany, in the last 10 years, up to 1% of the animals that were used for retina studies were used for gene therapy studies. So what we propose here is to use the retina chip for live screening of drug delivery and to collect the retinal organoids for histological evaluation of AAV cell tropism, which of the different cell type within the retinal organoid is being infected by each AAV. To mimic the subretinal injection, we had to change the chip design slightly, so we included an extra volume on top of the retinal compartment to assure that we have a controlled volume to deliver a defined concentration of AAV particles. We seeded the RPE 14 days before the organoid to assure that we have a tight and well-differentiated RPE layer. The organoid was then embedded within the hydrogel, and after hydrogel cross-link, we added the drug on top. The drug here are AAV capsids, which contain the genetic material for GFP. So when these viral particles infect a cell, the genetic material in the cell nucleus will be using the cell machinery to produce GFP, and they will be then fluorescently green. Before adding the organoids, we did a small validation test with RPE only, with and without hydrogel, and we added the viral particles to understand if the barrier is tight. And indeed it was. After 1 week of drug treatment, we could have a recovery of 92% in the subretinal region and less than 1% recovery in the media flow. So there was also minimum hydrogel effect, retention effect. Here you see how the RPEs look after 1 week. So they were actually infected, as we see on GFP, and they were a tight homogeneous layer in the whole retinal compartment. So next step was, would these AAVs affect the organoid as well? So we use semi-quantitative image analysis. We could define the region of the retinal organoid and the region of RPE only. And first we use conventional single-stranded AAV vectors and evaluate their capacity to produce GFP exposure over time. And as you see, up to 3 weeks in culture, there was still an increase of GFP production. Next step, we moved to double-stranded AAV vectors. They have the advantage of being already having already double DNA channel molecules, so the onset of fluorescence will be earlier, so it's a little bit more efficient. The only limitation is that the size of the gene that you can put inside of a virus is a little bit smaller. And here we tested 4 different AAVs, AAV2, 9, 7M8, and AAV10, in 3 different organoid ages, the 80, 200, and 300. And here there's a main difference, which is younger organoids have a higher density of ganglion cells, while older organoids, in older organoids, the ganglion cells degenerate a bit, and it can happen they are very difficult to detect. What we observed was that AAVs were able to infect all cell types. Organoid age did not matter much, but we've seen a clear dose response with 3 different AAV concentrations, and the system was sensitive to detect the different inefficiency of each AAVs. And I would here highlight the most efficient one was 7M8, which is indeed already in clinical trials for AMD therapies. This data was also confirmed only on the organoids by gene expression, so we quantified GFP on the organoids, and this was much higher also in AAV7M8 as in the other AAVs. So next step would be to evaluate the cell tropism. After doing the screening and the data that I just showed you, we collected the organoids and did histological analysis for cones, rods, Muller glia, as well as ganglion cells, amacrine, horizontal cells. Cone, rods, and Muller glia were the most strongly infected by all AAVs. 2 important species-specific observations that we got from this study was AAV9 efficiency was much lower for every cell type, which is reported to be very efficient on the infection of rodents, but apparently is not on humans, and this has been observed by other groups also on iPS-derived retinal organoids. Second observation is that AAV10 is known to be Muller glia specific in rodents, but we show here that they are also able to infect human ganglion, iPS-derived ganglion cells, as well as horizontal cells, and this is the first time that it was reported in the literature. So now we are ready to test new generation AAVs. These new generation AAVs were obtained from a random peptide display library using AAV capsid as a scaffold, and then the whole library of these AAVs was injected in the mice, and after 24 hours the mice was sacrificed, the retina of the mice was collected for DNA analysis, and they were able to identify which AAVs were there in a higher concentration using some kind of DNA barcoding, which it's definitely not my field, so this is everything I know about it. Then they identified AAV2NN and AAV2GL as being the most potent, so we compared them on a chip with our most potent AAV until now, which was the 7M8. And both showed to be superior on transducing rods, cones, and Muller glia, superior to 7M8, and most importantly selective. So there was no transduction of ganglion cells, amacrine cells, and horizontal cells. So we could consider that we validated our data against specific in vivo data from mice, dogs, and non-human primates, and human explants. Here you can see on the top, this is a human explant, and we see that both AAVs are mostly on the photoreceptors, and here on the bottom we see after subretinal injection in mice that the retinal tissue is well transduced, and both AAVs are also superior to 7M8. When we compare this with our data, what do we see? In the retina chip, it's more than 3-fold efficient NN is more than threefold more efficient than 7M8, which recapitulates exactly the threefold that they observed in the in vivo mice subretinal injection data. What we were not able to really recapitulate exactly was that the GLAAV in the retina chip is closer to the in vivo data when they use a promoter that is specific for photoreceptors. So in all of our assays and in this in vivo study, they use CMV as promoter, which is supposed to work theoretically in every cell type except some central nervous system cells. So this can indicate a possible interspecies difference that definitely needs further investigation. So to conclude, the retina chip allows a unique RPE photoreceptor interaction suitable for drug toxicity and drug delivery monitoring up to 21 days. Suitable for systemic and subretinal drug delivery, intravitreal will be a very important next step. Allows to study human-specific tropism of recombinant AAVs, and more important for screening, that it's difficult to do in animal models. And we could validate next-generation AAVs against human ex vivo and mouse in vivo data. So the retina on a chip platform is a drug development asset for drug development, where it's possible to get mechanistic insights within an organized 3D tissue. I would like to thank all of the people involved in this study, especially the microorganel lab, to Peter Loskill and my colleagues there, to the Stefan Liebau group in Tübingen, and our colleagues in Bochum-Hingelheim, Matthias Lux and Christian Schon. Before I'm happy to get your questions, I would like to catch your attention for the EUROX conference that will be within 1 month in Grenoble. It's the conference of the European Organ-on-Chip Society, and the registration is open until the 15th of June. Thank you.